Good morning. My name is Helen Shum at the University of Washington, and this is our next in the series of our viruses and vaccines seminar. So today I am delighted to welcome JT McCrone, who is going to be joining us here at the University of Washington, or actually at the Fred Hutchinson in May of this year as a new assistant professor. So JT will be giving us a talk entitled "What Lies Beneath Large-Scale Phylogenetics and the Processes That Drive Epidemic Waves." And then we'll also be providing our SARS-CoV-2 genomics update. So JT did his doctoral training at University of Michigan, followed by a postdoc at the University of Edinburgh. And he's currently employed at Helix, which is a biotech company that focuses on population genomics and genomic epi. And today, JT will be talking about his work throughout the pandemic using phylogenetic approaches and large data sets to characterize the geographic spread of SARS-CoV-2 in and between communities. So thank you so much for joining us today, and welcome to Seattle.
Thank you. Thank you for having me. So we'll start with the weather report, if that's all right, or the COVID genomics update. Here we are. So I guess I have two roles today. I'll give the talk and then also giving the update. So I know last time we talked, cases were trending downward in the United States, and that's still the case. So this is data coming from the CDC COVID data tracker. And we can see here the cases in blue over time plotted against the testing positivity in orange. And so most of these metrics have been trending down since, the start of spring. Here in blue when we had a little peak. I think if we look across the country too, at the community level, this is a metric that the CDC calculates based on hospital admissions and hospital bed usage. Most of the counties in the country, right, are in this low demographic of limited community transmission. So this all sort of trends with the fact that there are a decreasing number of cases in the country. I think even given changes in testing behavior over time, that's probably a trend that's true. If we look then at wastewater, this is data from BioBot. Again, we see the same story. So here, each region of the US is plotted in a different color, and then the national average is the blue dashed line. And so here you can see again, similar trend, wastewater levels of SARS-CoV-2 have been decreasing again since about the turn of the year. So we're getting multiple metrics showing the same story, right? That cases are going down. We've made it through the winter wave, essentially. Looking at variants, XBB.1.5 has been steadily growing in the US over the past number of weeks. It still remains the dominant variant. There's been some interest lately of a couple new XBB subvariants. So we have XBB.1.9 has been broken out on the CDC COVID data tracker. They break out variants once they reach above 1% at the national level. So there's been some interest in XBB.9 and its subvariants, but really a lot of talk is around this XBB.1.16 subvariant, which the nowcast model the CDC has at about 7% across the country. I think the last measurement was closer to 1%. That's a few weeks ago. So extrapolating out using their model, we expect that it's around 7%, 5 to 10% perhaps around the country. Now XBB.1.16, focusing here, is similar to XBB.1.5 antigenically. But it does have two mutations in the spike protein, one in the nucleotide N-terminal domain here, E180V, and then one in the receptor binding domain, 479 or 478. It's fairly spread evenly throughout the country, so we're seeing it in multiple regions at similar frequencies, unlike XBB.1.5, which began on the East Coast and then spread out throughout the nation. And one of the reasons that we're interested in this variant, I suppose, is because it's risen very rapidly in frequency in India. So it's now the dominant variant in India, present at about 100%. This is data coming from Pub Spectrum, which is pulling data in from GISAID. And India has seen a rise in cases recently. So this is shown over here from the Outbreak India dashboard. And so the rise is not, it's not as high as what they saw last summer. But it is still quite a substantially steep increase in cases. And so this is something that I think the WHO has declared a variant under moderation or under monitoring. something to keep an eye on as it goes. There was a recent preprint on this variant that showed that it had similar antigenicity as XBB.1.5. And so the hypothesis is that this is spreading more rapidly, not because of sort of antigenic escape, but perhaps it has a transmission advantage. Although I haven't seen any data to suggest that, other than the fact that it's increasing in frequency in multiple locations.
Hey, JT.
Yeah.
This is Allie. I probably should know this, but I don't. And so I'm wondering if you do. I'm wondering, do you know if India has like the same sort of access to antigen tests as we do? Like, or should, I mean, like, I'm still expecting sort of like differential reporting and things like that, but would we expect more PCR-based reporting and sort of, yeah, I guess like how might we expect these curves to look differently in India than here, like, even in the absence of like different variant dynamics?
Yeah, I don't have a good sense of how to compare like apples to apples Indian case reporting with case reporting in the US. I know, looking at other data sources like Our World in Data and even the India dashboard across the whole pandemic, this recent rise is very small compared to what they saw with Delta and Omicron. I don't know how that, I don't know, the absolute size of the cases will be affected by testing behavior. But I would expect maybe going back to, March, that testing behavior would probably be similar. And so I think maybe the steepness of the curve is maybe what's more striking than the absolute number. Yeah, but I don't know how to compare, those sort of biases. Yeah.
Okay. Yeah, that makes a lot of sense. Thank you.
You're welcome. All right. Now for the feature presentation. Okay, so as Helen said, I'm currently a senior scientist at Helix. Helix is a population genomics company. They do a lot of sequencing work on human exomes as well as viral surveillance. I've worked in viral surveillance for just under a year, but I spent most of the pandemic as a postdoc with Andrew and Bo at the University of Edinburgh. And so this talk will try to bridge my time, but we'll draw heavily on my experience in the UK during the pandemic. So we typically think of epidemics as a branching process where cases infect other cases subsequently down over time. But when we model this branching process, either explicitly or implicitly, we're not usually modeling the branching process as it is, because the data that we have doesn't represent the branching process. The data that we have is usually case counts over time, right? And so while our models account for these interactions, the data don't. And so often we're limited in what we can say about the data. And so in the simplest case, a simple SIR model, right, we get an epidemic trajectory that looks like a very nice curve. And as we know from the pandemic, the case trajectories that we're often working with don't follow these nice trends, because we have differences in human mobility patterns, differences in social mixing, as well as virus evolution. And so, right, epidemiological processes are complex, and often we reduce them to simply looking at cases as if every case is the same. But they're not the same, right? Cases differ by location, they differ by patterns of behavior, and they differ by variants. And so when we group them all together as this blue color, we miss out on the underlying processes that are driving the transmission patterns within the population. And so ultimately what we'd like to do is enrich our understanding of epidemiological processes by explicitly accounting for this branching process that's underlying the data. And phylogenetics allows us to do that, because when we sequence cases, we can estimate the relationship between cases using the similarities within the genetic code of the virus, and that approximates this ancestral reconstruction, right? And so this is something that people have been using phylogenetics and phylodynamic models for well before the pandemic, but it's something that's really been useful, I think, during the pandemic, as the data sets have grown. So before we get into some of the pandemic data, I think it's useful to see this process and this way of thinking in action. And a good example of this is the 2018 and 2020 North Kivu Ebola epidemic. So this was a an Ebola outbreak in the North DRC, shown here on the map. It's the second largest Ebola outbreak to date, second only to the West African outbreak, which was about 10 times this size. Most Ebola outbreaks are a couple hundred cases. The West African outbreak was near 30,000 cases. This one was fairly large, so with about 3,000 cases. And so we were working with some collaborators in the DRC who were doing sequencing there. I know some people on this call were also involved in this analysis. And we in Edinburgh were providing some phylogenetic analyses to get some insights into what's causing these cases and the trajectories that we're seeing. If you see the reported cases over time for this outbreak, you can see it's not a standard, right, SIR simple curve. We get cases that are maintained at a pretty constant level for most of the outbreak before increasing slightly, but then again maintaining at about a baseline. And so it's very odd for epidemics to maintain a flat level. Usually epidemics are either growing or shrinking. They don't usually have a rate of change of 0. And so this is a great example of how sequencing and phylogenetics can help us understand what's underlying these epidemic waves. So here's a phylogenetic tree of about 350 samples taken throughout the course of the Ebola outbreak. Samples here are shown in circles, and they're colored by health zones. So this is similar to what we might think of like a county or zip code demographic in the US. And then we've inferred along this phylogenetic tree the ancestral location of cases, and then colored them as well. And so you can see the outbreak started here in this green region. This is a health zone called Mabalako. We had 28 samples, and it circulated here in 2 distinct clades. It then moved through 2 distinct events into the blue zone here called Beni, which led to onward transmission both in Beni as well as the rest of the pandemic, or the epidemic, excuse me. And so we're able to see here that at the population level, we had cases that were similar to a flat line, but really this is because we're moving between different communities. And often communities are being infected by multiple transmission chains at the same time. And we can really get a sense of that if we take this tree and break it apart as the virus moves from region to region. So we'll cut the tree at each of these inferred exportation and importation events, and then plot those transmission chains alongside the cases in each location. So that's been done here. You can see now we're showing the tree with cases superimposed in the background. So here, this was that initial outbreak in Mabalako, accounting for cases in early August, as you can see here. And then remember we had the virus jumped from Mabalako into Beni. That happened here. And that happened twice. So I guess it actually happened a couple of times. There's a couple of hidden ones here. And so then the cases in Beni, which spanned from August into November, here were caused by these overlapping transmission chains in the community. So these are 2 distinct importation events spreading in the community, causing seemingly one epidemic trajectory. The virus then left Beni for a period of time, and then we see no sequences and very few reported cases in this inner period. But then it came back as one transmission chain here in, I guess, mid-April. We can compare this to dynamics in another health zone, Butembo, where we see a fairly constant level of cases over the course of the epidemic. And the phylogenetic results suggest that there's limited local transmission. So you can see we have a lot of imports shown here that lead to just singleton cases or just a small cluster of cases, but they're not spreading as a growing epidemic as we saw in Beni and Mabalako at the beginning. And so here we can also see that many of these importations are coming from the same location in Katwa up above. And if we were to look on a map, we could see that Butembo sort of wraps around Katwa. And so we have a very large, I guess, epidemic in Katwa that's routinely spilling over into Butembo, but we're not getting a lot of onward transmission to Butembo. And so I think this is a very powerful approach that allows us to at least descriptively get a sense of what's going on in the epidemic, what's causing the epidemic. And in this case, it was being caused by a high level of mobility between different communities, followed by subsequent spread in those communities with overlapping transmission chains, leading to sort of the rough epidemic curves that we were observing. at the population level. And so I think this is a very nice descriptive study to get a sense of how we can use phylogenetics to describe transmission chains and transmission lineages within and between communities. But what we really want to get at is a quantitative characterization of, are there differences between transmission lineages? Do they contain mutations that are more likely to spread faster in a community? One of these lineages did have a mutation in, Ebola's equivalent of spike. That was observed, or mutation like it was observed in the West African outbreak. And so, there's a hypothesis that this is, an adaptation to human transmission, but because there's limited data, it's very hard to tease that apart and distinguish, viral factors from local factors. Was there just more mixing in certain locations? So these are really interesting questions to answer. They're important questions to answer to understand virus transmission. But historically, they've been hard to answer because even though the data is very powerful, there's a lot of uncertainty in these reconstructions. And we ultimately don't have very much data to answer these questions. And so, historically, we've been tasked with building models that can grapple with limited data and be informed by multiple data sources to try to tease apart these really complex questions. And we've been faced with a totally different problem during the pandemic, right? So in the past, we've been faced with limited data, and we've needed sort of strong, heavy models to get us over that hump. But in the pandemic, we've been overwhelmed with data, right? There's been a huge sequencing effort globally to track the virus's spread within countries, between countries, and its evolution. So we've been overwhelmed with data, and that data has overwhelmed our traditional models. But the questions remain the same. And so we've been tasked now with, instead of building models to handle small amounts of data, and so to get the most out of small amounts of data, we're now, we can build simpler models and rely on the richness of the data, but now we need models that are computationally tractable in this world of large data sets. And so some of the characteristics that we can use to our advantage in this case, and these have been noted by really anyone who's doing sort of large-scale phylogenetics of COVID. We have well-defined large-scale clades, right? So this is a tree from Coffee Tree showing most of the samples from the pandemic, most of the clades. I think it's missing the A lineage that was around early in the pandemic. But this is most of the B lineage. Here we have the Delta wave up here. This would be Omicron now down at the bottom. This was the first variant of concern, Alpha B117. And so we have very well-defined clades at a large structure. However, within each clade, you can see we have very poor structure. And we have a lot of polytomies where the ancestral relationship between all of these subclades is uncertain, right? And that's why they appear on the tree as coming out of one single ancestral node. So there's some relationship between all of these different colors. There's some probably distinct relationship, parent-children, all the way back to this ancestral node, but we can't estimate it using the sequence data because the virus is spreading so much faster than it's mutating. And also we're faced with another challenge where we have many short branches. So most of the branches in this tree, Omicron, excluding Omicron, these long Omicron branches, most of the branches contain no mutations, one or a handful of mutations. And so what this means is that we can leverage some of these characteristics to make our normal analyses more tractable. Because there's significant uncertainty in the tree, and because I was training with Andrew at the University of Edinburgh, and when you have a hammer, everything looks like a nail, and we took a Bayesian approach, because a Bayesian approach allows us to naturally account for uncertainty in the topology of the tree. But the problem with that is that Bayesian approaches scale poorly, right? And they do so because although we're integrating over the uncertainty in the tree, the number of trees we have to calculate to do that is huge. So the number of trees available, for even a data set of more than 50 samples, I believe there's more possible trees than there are particles in the universe. So we have this gigantic space that we're trying to move over during this analysis. To compound that problem, we move from tree to tree in very small steps. So if we're analyzing one tree, and then we're going to entertain another tree, usually the new tree that we're proposing is very similar to the past tree. And so we have a huge space to explore, and we have to take very small steps to explore it. Further, each step is very computationally expensive because we're calculating these very large likelihood functions over huge data sets. And we're also trying to manage those huge data sets in memory at the same time. And so this is akin to like exploring the Alps with a very heavy backpack, but you can only take, like a tiny, maybe not even a full step at a time. And so essentially it's an impossible task to do in its fullest sense. And so what we did first was to limit the number of trees that we'll entertain. And so we use the fact that most of the trees most of the clade structures in the tree are well defined. And so we limited our search to reasonable trees. So we're going to keep these large clades, right? The alpha clade, the delta clade, the omicron clade. Those are fixed in our analysis. And we only entertain trees that respect those clades. So here we have a blue clade and a red clade. As we're going to integrate over all possible trees, we only accept trees, or we only entertain trees that respect the blue and the red clade. Right? We never propose a red tip mixes with the blue clade, right? Because we believe essentially the underlying structure of the tree, the large scale tree. Okay, well that limits tree space a little bit, but we're still exploring it in small steps, and each step is still computationally expensive. One way to get around the computationally expensive part is to simplify the calculations that we're doing in the analysis. So usually in a Bayesian phylogenetic analysis, we have our data, which is sequence data, so a large sequence alignment. It can also include geographic data of where the samples are taken. And we're interested in calculating the probability of this, of some parameters given this data. I think I have that backwards. At any rate, that's proportional, to some tree that we'll be exploring, and some the likelihood of some tree given the data, and some model of sequence evolution. So that's here some model of how A turns to T or G or C. We also have the same tree and the same geographic data now, but some model of how viruses spread around the country. And then because this is a Bayesian approach, we have to include our priors, which account for our prior belief in these parameters before looking at any data. We then have, we search over all this parameter space and sort of chart out the possible trees and parameters that make sense. But this sequence likelihood ends up being a very long equation. And this data here, this alignment for this case of SARS-CoV-2 is a very big alignment. So it takes up a lot of memory. And so what we did is we used the fact that most branches only contain one or a handful of mutations and simplified the likelihood. So instead of accounting for every possible change that could occur along a branch, we assumed, we're sampling so densely that we don't have any changes that occur without us seeing. And so we don't have to account for every possible change along a branch. We can just account for the changes that we do see. And that means that we can assign mutations to branches and then calculate what's the probability that we see, 3 mutations along a branch, instead of calculating what's the probability that we see 3 mutations along a branch, given that there could have been any number of mutations along that branch. And then we also introduced new data structures. So instead of storing the alignment in memory, we're now storing just the number of mutations along a branch. And that's a much more efficient way to store essentially the same data. We can look at the amount of time it takes to calculate the likelihood of a full data set, the full tree. And you can see here the classic way in blue scales linearly, but scales very, very rapidly. So this is the amount of time it would take to recalculate the full likelihood in seconds. And if we're doing millions of these in an analysis, you can see how this would be a problem to do anything in a reasonable amount of time. On the other hand, when we're just using this Poisson method of counting the number of mutations along a branch, again, that scales linearly, but at a much lower rate. And so it means that doing these large-scale analyses becomes feasible. Also, when we have a huge alignment and we're doing a traditional analysis, that to make that possible, that data set needs to fit on a GPU. But again, it takes up a lot of memory. And so this is the memory usage of a classic analysis plotted against typical GPUs that are out there that an academic lab might have access to. And so you can see that very quickly, we run out of room for running these analyses. However, with the new data structures, again, storing mutations in each branch, that scales much more reasonably. And so now we can run these analyses up to tens of thousands of samples on a laptop at home. I would say probably up to 10,000 samples on a laptop at home. This is still a large scale analysis that takes some time to set up, but it becomes feasible. Okay, so we fixed a couple of these problems, right? We're still exploring tree space in very small steps, but we've limited the number of trees that we're entertaining, and we've made each step computationally tractable. So our first application of some of these models was to describe the initial wave in the UK. And so this would be the wave 1 in the UK. We wanted to estimate the number of importations over time into the UK, and then we wanted to follow the history of those importations in the community throughout the course of the first wave. So when we talk about lineages, in this case, we'll be talking about transmission lineages. These are distinct from variants of concern or pangolin lineages. A lineage in this case is an introduction into the UK, followed by subsequent spread and detection. This would be an introduction into the UK, followed by a single sample. And then, of course, there will be unobserved introductions into the UK. And so we expect that our calculations here will be sort of a lower bound on the number of true importations into the UK. This analysis was done with about 50,000 sequences, it's very hard with a data set of that size to do a full geographic model accounting for all possible transitions along the tree from each country. And so what we did is we simplified it. We had the UK shown here in blue and the rest of the world shown here in purple. And we just estimated the introductions from the global population into the UK. We then combined that with a model that accounted for air travel over time to assign a source location for each introduction probabilistically. So we used the phylogenetic approach to give us our when introductions happened and what cases each introduction led to. And then we used an epidemiological model to assign, given that this introduction happened on this day, we look at the number of cases in each country and then travel to the UK to probabilistically assign where that introduction came from. So the first wave in the UK ran from January 2019 to about the end of April 2020. In the middle of March is when the UK went into a lockdown, just to give you a frame of reference for what's going on during this wave. Here we've plotted the number of introductions per day into the UK and then colored them by the size of those onward transmission chains. So you can see we have a couple introductions that are very large, that make up over 1,000 genomes. Those tend to occur early, right? So we have most of our red and blue introductions, which are the large ones, occur early in the epidemic. This is when the population is most open and transmission occurs. Okay, so we've plotted the number of introductions colored by their size and noted that earlier introductions tend to be larger. We've also looked at where those introductions are coming from. So early introductions originally tend to come from China because this is where most of the cases are coming from, right? That then switches in February to most introductions coming from Italy. If you remember, Italy had a very large outbreak early in the pandemic, and then it moves to Spain and then later on France in blue. So we have this dynamic source of importations changing over time. And you can begin to understand why it would be so hard, right, to put in place targeted travel restrictions from different countries. Next, we looked at, okay, once those introductions are in the UK, what happens to them? Where do they go? How quickly do they grow? And so here we've plotted the number of sequences in a location versus the number of lineages in that location. As you can see, like the more sequences, which is here proportional to the number of cases in a location, the more introductions have ended up in that location. So areas with very high numbers of cases in the first wave also received a very high number of imports. Or a very high number of transmission lineages that spread within those countries, those counties. If we look now at each transmission lineages, we see that the larger the transmission lineage is, so the higher the number of sequences in that transmission lineage, the more widespread it is. So this is plotting the number of sequences in a lineage versus the number of administrative regions or counties that lineage has gone to. So large lineages tend to be widespread. Regions with a high number of cases can have a high number of transmission lineages in them. And so we're getting this picture of a very open country with a lot of movement between regions with very easy transmission dynamics between geographically distinct regions. We can look at each lineage, and here's a couple examples, and plot where they were observed over in the UK. And so here, each of these is an introduction into the UK, followed by subsequent spread. We're plotting how many cases of that lineage are found in each location by the size of the circle. And then the color represents the earliest time that lineage was detected. So light colors would be where the lineage was initially detected. Dark colors represent where the lineage was detected later. And then here we're plotting the pairwise distance between samples. And so here you can see lineage 1 was a very widespread lineage. It was found throughout the country. It was found concentrated in a couple of regions, right? That's why we see a peak of distances at 0, probably located here in the Midlands, but otherwise fairly well mixed throughout the country. We see something similar with lineage 2, again spread wide throughout the country, fairly evenly, right, with a much flatter distance here. Lineage 3, although it was found in different parts of England, it was mostly concentrated on the west coast of Scotland. So that's shown here. So we're getting some geographically distinct transmission patterns in different communities. We also looked at, in addition to the spatial dynamics of introductions once they enter the UK, we looked at the temporal dynamics. So how often are we detecting new lineages, and how often are they going extinct? And so that's shown here in red and blue. So red is the number of introductions per genome sequence. And you can see early in the epidemic or in the pandemic, it's very flat. It's relatively flat. So we're getting, a constant amount of introductions over time. And then when the UK goes into lockdown, now we're no longer detecting new lineages as often, but each subsequent sample, right, is a representative of an introduction that we've seen prior. And so that's why this red line is decreasing. The flip side is how often do these transmission chains go extinct? And so that's shown in blue, where we have the rate at which lineages are no longer observed for 4 weeks. As you can see, the lockdown begins in March here, and then we start to see these introductions go extinct. As the cases come down, right, smaller introductions tend to go extinct faster. But eventually, we lead to a high, we have a low number of cases, and most of the introductions have gone extinct. So in summary of wave 1, it was triggered by well over 1,000 introductions into the UK. The size of the change followed a power law, where a high proportion of cases came from a small number of introductions. The sources over time were dynamic, as we saw it move from China to Italy to Spain to France. And then as it comes out as a surprise to us now, lockdown measures were effective in reducing both the number of cases, but also the number and size of the transmission chains spreading around the country. So this was a fairly qualitative description of these transmission chains within the UK. And we wanted to move towards a more quantitative understanding of how human mobility and travel restrictions affect onward transmission. An example of this in practice was an analysis we did looking at the rise of Delta within the UK. So Delta, as you remember, was a variant of concern that originated in India. It then spread around the world and spread very quickly within England and outcompeted Alpha, which was the reigning champion in England at the time. This is a tree, just to give you a frame of reference, showing the rise of Delta first in India in blue, and then its subsequent spread around the rest of the world in yellow, but more specifically, it spread in England in red. This plot here is showing the frequency of Delta in both India over time versus England. And so you can see that these curves follow each other very closely. The solid lines represent the proportion of cases sequenced. And so this is important when we're understanding movements from one community to another. So the UK had a much higher proportion of cases sequenced over time. Now for this analysis, we didn't have that very rich epidemiological data set that we had before, which allowed us to assign locations to incoming travelers based on travel history data. But we did have anonymized mobile phone relative movements. And so we could say from week to week, what's the relative proportion of travels from India to the UK. And so what we did is we took a phylogenetic approach to estimate imports from India into England. And then we compared that with this estimated importation intensity using aggregate mobile phone data and the number of cases in India. We can see here, this is plotting the number of introductions per day from India into the UK over time. So again, this is based on the sequence data. And then we can compare that with now this relative normalized estimate of how many we should expect relative to the previous time period using, again, aggregate mobile phone data. And that's shown in this dark line in blue. The gray bars here indicate when the UK put India on the red list, which meant that incoming travelers were under mandatory hotel quarantine. and when that practice went into effect. So it was announced a few days before it was put into effect. That's shown here. What you can see is up to the announcement of India going to be on the red list, we had a very well correlation between the number of phylogenetic imports over time versus the expected imports over time. Remember, this is a normalized measurement, so we don't necessarily expect it to fall right within the bounds. But you can see we get a very high correlation, so the R-squared value here is very high. However, after India goes on the red list, you can see these two metrics become decoupled. Right, so initially, there's very few travelers from India to the UK, and that plummets the number of expected importations, and then it rebounds as the number of cases in India skyrockets in the spring of 2021. We don't see similar dynamics from our phylogenetic data set. Instead, we see the slow decline and then a steady decline after that. And so, right, whereas before we had correlations between imports and human behavior, after the red listing of India, we no longer see that correlation. So that suggests either, right, the mandatory quarantine is working, and we're now decoupling imports from cases that are entering from India, or there's some behavioral change in individuals who are traveling to the UK. Right, so it's quite possible now that there's a red list, you're less likely to travel. If you're feeling sick, also in India, you're probably less likely to travel than perhaps you were before. We can compare this as sort of an internal control with the number of imports that are coming into the UK from the rest of the world. And so that's shown here in yellow. As you can see, as our number of imports coming from India decreases, the number of imports we're getting from the rest of the world increases, right? Because these cases are going from India to the rest of the world and then coming into the UK. And we can see that over the study period, we maintain this high level of correlation between expected imports from the rest of the world and phylogenetically estimated imports from the rest of the world. And so this suggests that the travel restrictions put in place against India, while effective in limiting the travel and the onward transmission into the UK, were not effective in limiting, the spread of the virus because it was too little, too late, essentially. Another way to look at this is the number of singletons. So if an import comes in and it leads to onward cases and we sample it multiple times, that suggests that it's a higher level of transmission. Whereas if an import comes in and we only sample it once, suggests that that's a low level of transmission. So the proportion of singletons we're seeing over time is also some indication of how well these restrictions are put in place. You can see the proportion of singletons before and after the lockdown went into effect is fairly constant for people coming from outside of India, whereas we see this decrease for people coming from India, which again suggests that the travel restrictions put in place are effective at limiting introductions from India. We can also look at what happens to these introductions once they enter the UK. So here are three large transmission lineages. These plots movement moves in a counterclockwise direction. So both of these are early introductions into the northwest of England, the greater Manchester area, with exports both to the south and to the north. So these are moving down and up. This is an introduction into London, which then was spread, throughout the country with limited onward transmission at the rest of the country. This is different than what we saw and then west of England and India. And so this is just highlighting that, human patterns of behavioral change, affect how these variants spread within the country. Over the course of the rise of Delta in the UK was easing out of lockdown, of its awful lockdown. And so we put travel restrictions on travelers coming from India. However, local movements increased throughout this time period. And so we can use that to understand how far the virus is transmitting within the country and how different lockdown procedures affect that movement. So this is showing the proportion of movements within the UK, phylogenetically inferred along the tree, the sort of a CDF of that. And so here you can see under lockdown, a high proportion of movements were very small. So about 80% of all movements were less than 100 kilometers. But as we ease out of lockdown in yellow and then are fully open in red, we get this longer tail. So a higher proportion of movements are now going 200, 300 kilometers. So this is just indicating that as we come out of lockdown, as there's more human mobility, we're seeing that mobility manifested in larger range transmission within the country. We also asked what amount of local mixing, how does local mixing affect the rate at which Delta reached 100% in a given county? And so here we've binned counties by their amount of mixing, human mobility at the local level, and then plotted the frequency of Delta over time. And you can see that areas in yellow that have very high levels of mixing, Delta rises faster. And so in this analysis, we were able to go a step further than we did in the first wave and begin to quantify how human mobility patterns affect both local transmission of Delta, but also imports of Delta into the country. And essentially, we learned from this analysis as well that it's very hard to have a targeted approach to limit imports into a country, right? Because the UK had a targeted approach against India. However, it was too little too late. And that targeted approach came on the back of opening up local restrictions. And so any imports that did make it into the UK, which are bound to get through, were coming into a very open society where transmission was readily available. Okay, so that covers, a large chunk of the pandemic and a large chunk of my experience of the pandemic in the UK. But more recently, I've been working at Helix, and Helix is focused on sort of two branches. One is human genomics and human exome sequencing, and the other has been viral sequencing. And it seems to me, as someone who did not work at Helix at the start, that viral surveillance started out because if you're used to swabbing individuals' cheeks and sequencing DNA, then you're probably very good also at swabbing individuals' cheeks and sequencing viral RNA. And in fact, Helix has been. And for a large part of the pandemic, they were running diagnostic tests for pharmacies. So they ran over 1 million diagnostic tests for pharmacies around the country. On the back of that pharmacological testing, they also sequenced a large number of samples for surveillance for the CDC. And so we've sequenced, about 400,000 isolates. About half of those have been as sort of a sequence as a service that have been returned to individuals who, or not corporations, but researchers who were interested in sequencing. But the other half have been returned to the CDC and been made publicly available on GenBank. And so this represents a very large data set. And because it's coming from one data source, we also have very rich metadata. So we're able to do things that are very difficult to do within the US federated public health system, because we have rich metadata for tests coming from all over the country. And so ideally, I wanted to present a phylogenetic reconstruction of transmission in the US across the whole pandemic. And I wanted to do that so badly, but it has seemed like for the past month, it has been like both one step away and like an infinite step away. It's like Schrodinger's analysis. And so instead of preliminary analysis, specifically in California. And so this is an analysis that mimics what I presented before in wave 1 in the UK, where we'll be estimating imports into California and then looking at their subsequent spread over time. So the data that goes into this analysis is publicly available sequences from the Usher tree, and we'll be identifying introductions using a heuristic approach that the Usher team has also produced. We'll then augment that data with the rich metadata for samples that Helix has provided, and then we'll look at introductions and their spread for larger introductions. So this plot is showing introductions into California over time, colored by variant of concern. And then the x-axis is showing the sampling period. So here they're sorted by earliest and then latest sampling period. And so the rate at which we move down, right, indicates how quickly introductions are coming in. So we have a high number of Delta introductions coming in over a short period of time. And then that sort of flattens out. And then you can see here Omicron begins, and we have now a low number or a lower rate of introduction for Omicron. But also interesting, right, you can see as soon as Omicron becomes introduced into California, all of these Delta transmission chains that have been going on for months in some cases become extinct, right? Because Omicron's outcompeting Delta. And so this is nothing different than the frequency dynamics that we're all used to seeing, but I think it's interesting to see the effect of these variants on previous sort of transmission chains in different communities. We can plot the rate of import versus extinction over time. And I think that gives us some indication of how human mobility has changed throughout this period of the pandemic. So early on, you can see we have a fairly constant number of imports to export rate. And so we're getting, turnover happening very quickly. This is probably a lot of small imports that are going extinct over time. Right when Delta begins, right, we can see we have a spike in imports. We get a high number of Delta imports from other states as well as international. And then that's followed by a rise in the rate of extinction, right, as all of these previously circulating lineages go extinct. That's different than what we see with the Omicron wave. So here's the Omicron wave. We actually have a low number of imports at the start of the Omicron wave. And this could be for a couple of reasons. One, when you have a high number of genetically similar introductions, it's hard to distinguish each one. So that may be an artifact here, or it may be that individuals are changing their behavior because an rapidly increasing number of Omicron cases. But what's interesting is that even as the rate of import goes down, right, the rate of export or extinctions skyrockets, and that's because we had all of these Delta chains that have now gone extinct because Omicron's not competing them. And then as we move into the summer of 2022, you can see we get this rebound now where we have, again, this constant turnover of sort of well mixing between California and other states. The next question was, once something gets into California, what happens? And so this is showing a reconstruction of some of the larger transmission chains. And you can see the cases here are colored by date of sampling. So the light ones are early and the dark ones are late. We get a high number of early cases in Southern California, San Diego, and LA, a lot of mixing between Southern California, San Diego, and LA. And then Southern California is acting as a source of transmission around the rest of the state. So you can see we have a high number of exports out of Southern California up to this San Francisco Bay Area, with some reintroductions back into Southern California, but right, many more exports from Southern California than we're seeing exports from Northern California. And looking across all introductions, we can plot the relative contribution of each county to cases in a local area. And so, I don't know, as a Midwesterner, I have often thought of California as like the crust of the US, but looking at it on the map, it's actually a very north-south state. And so in this matrix here, we plotted counties by their, from north to south, which makes it a very nice introduction. It would be very difficult to do this for something like Tennessee. That's a very east-west state. But you can see here, right, this is Southern California. We get a lot of mixing within Southern California itself. And so it forms this block. But then you can also see that this block extends into northern counties, much more than we see sort of up here. And so this is, again, just showing what we had a sense for up here is that the South, at least for parts of the Omicron wave, was acting as a main hub of transmission around the state. So this has been, to summarize, this has been a whole lot of work spanning multiple years of the pandemic, relying on a whole lot of data providers and a lot of sequencing. And I know that there's been talk now as we're coming, funding for those institutions is uncertain. How can we leverage the work that we've developed over the course of the pandemic to give us a better handle on viral surveillance in the future? And so this is something that Helix is actively interested in. But I think it's also interesting to think about, we've built up this infrastructure, but we've also built up a very rich data set. And how can we use this data set to understand viral surveillance of other viruses? And so something that's interesting is that Helix uses a hybrid capture sequencing approach. And so we often capture sequences of non-SARS-CoV-2 viruses as co-infections. And an example of this is our endemic human coronaviruses. So this is showing, just very quickly, the number of co-infections of SARS-CoV-2 with other endemic coronaviruses over time. And even though these are, right, this will be biased by both, the prevalence of these species and also COVID in general, these trajectories match what we see at the national level. But again, right, we don't know a whole lot about the transmission patterns of human endemic coronaviruses. I think probably everyone here on this call knows very close to the same amount as each other. That's, we don't know very much if we add it all together. But I think it's a very interesting question, because if we look, where do these sequences cluster for one of these species, so OC43, which is right here, we can see, they fall into a couple of distinct clades, and they fall into clades that have been distinct for well before, the pandemic started. And so while SARS-CoV-2 and other viruses move all around the country and all around the world, have these viruses been staying in the same place over time? It's very hard to tell, right? Because we just have samples from essentially North America, from the US during this time period. So we don't know, and it's unlikely that we'll get data to fill this in. But we do now have a very rich data set of human movement, and in particular human movement that contributes to coronavirus transmission, because we have this very rich pandemic data set. And so I think it's interesting thinking about how can we leverage what we've learned from SARS-CoV-2 patterns of transmission to maybe begin to tease apart, what's the chance that these clades really have been in the US over the course of the pandemic, undetected versus spreading around the rest of the world like other viruses. So I don't know if that's a good idea or not, but I think it's fun to think about where we're going. And then just to say thank you to this, this is a huge amount of work, and I've been fortunate to play a small role in all of it, but it stands on the back of a large number of collaborators and a large number of data providers. So just to acknowledge and thank everyone who's been involved. And I think we have a couple minutes if there are any questions.
Great, thanks so much, JT. I think if others could just go ahead and ask their questions, either in the chat or you can just speak up. And perhaps while you're doing that, JT, I'll go ahead and share the screen for the next seminar series.
Yeah.
I have a question, JT. That was really interesting. Thank you. Could you re-explain for me the branch-based likelihoods? I got a little bit lost there.
Yeah, of course. And I'm sorry, I skipped over it. And then I think I re-referenced it with things that I hadn't explained. So I apologize for that. So usually when you calculate the likelihood of like a sequence alignment given a tree, the models to do that were developed, when the sequences that were available were like mitochondrial genomes from, like great apes or something like that. So we have evolution spanning millennia. And so the models account along each branch, you're accounting for the possibility of unobserved mutations. So you have mutations that go, you have a C that goes from a C to a T, maybe back to a C, and then it ends up as a G when you sample it or something like that. And so the models are very sophisticated in that they can account for all of those unobserved changes. And then they also include uncertainty at each internal node, because we don't know what the state of each site in the alignment is. But when we sample really densely, we're picking up about every mutation. We think we're picking up mutations as they occur in the pandemic, and we don't think that there's very much of a probability that there's been a mutation, an A went to a C and then back to an A. And so instead of doing this sophisticated model that accounts for all of these possible changes, we say, no, that branch has one mutation, and then we know the rate of evolution from one mutation. And so then that lets us calculate, about how long should that branch be in time. So we're calculating the time tree from this mutation tree, which then helps us put, viral spread in context of other things that are going on, like human movement patterns and lockdowns and things like that. So it's a shortcut for going from, the number of mutations along a branch to how long is that branch in time.
That makes sense. Thanks so much.
Hey JT, this is Ali. I have a follow-up question on that. Because I was just wondering how you implement this. So it sounds like you are you saying then that you would traverse the tree, observe which mutations occur, and kind of like build a well, you don't even need the substitution model because you're not modeling unobserved mutations. So like what?
No, it's really simple. It's like, I feel like it's like macaroni art. like I tried to find a picture of someone like sketching a fancy picture at a museum, and then you zoom in on it and it's like just macaroni art, because that's what it feels like compared to all of the sophisticated, Bayesian statistical models out there. So it's just, you traverse the tree and each branch has a number of mutations, and then the likelihood of that branch in time just comes from a Poisson distribution given the rate of evolution. So the evolutionary rate, you know about how much time that's been, and then you say what's the probability that we would observe one mutation given that the branch is this long, and then you fiddle with the branch lengths until as you sample over trees.
Okay, that makes a lot of sense. And so like, is this built into like beauty now? Can you be like, can you toggle on like, this is the like substitution model that I want to use is not actually a substitution model. I want to use this like Poisson, or is it like an XML chunk that you have
It's
to
an
like,
XML, it's an XML chunk at the moment, but I think there's been some work in beauty, and so it would be great if it ends up in beauty. It should end up in beauty. So
Okay,
yeah.
That's cool. Because I think this would be potentially really helpful in terms of thinking about how we would be able to use Beast in like a public health implementation type context, right? Is I think like we'd really want that. We're often doing, intensively sampled outbreaks where we wouldn't expect back mutation to happen at a meaningful level. And we also definitely need ways for like Bayesian approaches if we were going to use them, like they would just have to be less expensive. So yeah, we can follow up later, but I'd be really interested in like how we would implement this.
sure. Yeah, that'd be great.
Thanks.
Okay, I think we are at time. So thank you so much, JT, for doing both sections of the seminar today. So that was really outstanding. Just want to remind everyone our next seminar is on May 16th. We have Barney Graham coming. He was previously, as many of you know, the head of the VRC, the Vaccine Research Center at NIH, and he led the development of the SARS-CoV-2 pre-F vaccine, as well as the RSV pre-F vaccine. He's a friend and colleague of mine. He's kindly agreed to come and speak to us about RSV vaccine development. So it's quite timely. It'll be a great talk from the leader of the field. Hope all of you can join us.